Hello. Um, I thought I'd do a little video for y'all. I was drawing some cells for fun, and I figured I would share it with the class because I do feel bad that we didn't get to have a lecture, and we're kind of coming to a close on sort of my contributions to finding the knowledge and sharing it. And so I just thought I would give, since we didn't get to have a lecture on this past topic, I would just give like kind of an overall sort of like story of the course, which will include the next two papers as well. So this is kind of just like an overview of everything and kind of the story that we've all told together about these cells. And like I didn't really, so like let me just get in here. So like when I was planning this course out, I was like, okay, I want to start like at the plasma membrane because like this like little layer of lipids, like if you zoom way into this, like these lipids just got like these nutso tails, right? And they all have them like this. And these things are freaking going nuts and just bouncing around. Let's see if I can like Google like lipid dynamics membrane. Let me see. Simulation. Well, I kind of want you to see this. Phospholipid. This is not. What? Whoa. I don't know what I'm looking at, but I think it's cool. Molecular surface. Residue type. And this is a phospholipid bilayer, and these are residue types. Amino acids, yeah. Yeah, crystal structure, yeah. That's cool. Plasma membrane? Yeah, okay. Dude, oh, this is gonna be cool. Whoa. Okay, let's watch this hole get opened. All right, look at that! Uh, I mean, they're just freaking out. They're just going bananas. Look at these. Just dancing. So these things, let me zoom back out. So these are the lipid tails, and these are the lipid heads. And this thing is making a hole, so it's like a pore that's forming because of like electroporation. So that's what's going on here. Yeah, that's interesting. What? Okay, I'm gonna have to watch this video on my own time to really look at this and understand this. But now, I feel like I have a better understanding of membranes by watching this video, so I'm glad that we're here together to do this, learning together. That's really cool. Um, there's the URL if you want it. Okay, so now you get that. So I wanted to start at the membrane, and that's where like piezo came from. And piezo, if we remember, it's like this like gazebo looking kind of thing. Weep. Um, what? Okay. No, I don't want to path it. Thank you. Making it smaller. Look at this. I did it. Beautiful. Great. Okay, and then like little calcium ions coming through. Oh, calcium's obviously white. Little calcium ions come through. Okay. And tension opens this boy. open, and this is piezo. So that's where piezo came from. And piezo, a graduate student, Saranya, in the Miller lab that I worked in for my PhD work, um, she was interested in piezo and was studying its role. And I showed you those videos of calcium and rho that she generated. And so they're interested in it being a signal to repair cell-cell junctions. So that's where that just kind of came from. Okay, so that's it. And so then I was like, all right, well, now we're there. Now we kind of want to build out from that. So we focused on one single protein. So the next thing that we focused on was like, you know, I'm a big actin cytoskeleton person, so we're going to be focusing a lot on the actin cytoskeleton. So let's just do an actin cytoskeleton paper. What's something that's really cool and interesting? Well, oh yeah, optogenetics is such a fascinating science tool. The way that we manipulated, you know, plant proteins to, you know, recruit different components so that we can just like, you know, activate a GTPase wherever we want. That's such a fascinating thing to me. As soon as I saw somebody did do that in like 2015 at a meeting, I was like, we gotta do this in the lab. And then I tried it. I got the proteins and I cloned them. And I tried it once and it didn't work right away. And I was like, yeah, I can't do this. I gotta finish my PhD. I can't start this other project right now. And so then I put it on the burner, but then back burner, but then another graduate student picked it up and she's got it working. And it's so cool. And other people got it working in tissues too. So up to that point, people only got it working in single cells. So we looked at the paper on stress fibers. So we went from this single protein of piezo into, you know, stress fibers, into a larger structure, stress fibers, which are made up of all different kinds of things. So I've drawn there the actin, but there's also, let's see, there's little myosins, of course, which are all over this. It's nice and beautiful. Look at them. Look how cute they are. Myosins are cute. So now we're looking at not just one protein, but we're looking at a bunch of proteins. The protein that the paper was concerned with was this protein called zixin, which I don't really know what it looks like, but I'm gonna guess it looks something like this. It's kind of like a long thing with like multiple domains, and it was recruited to, let's see, I don't really exactly remember. It was near the sites down here, so now I'll just kind of like draw them like this. So it's kind of like, you know, down here at these sites, like this, and then it was right where they activated. So like if they activated like right around here. And there's all kinds of other proteins. There's little scaffold proteins that like go this way, you know, and connect these filaments like this. That help organize it into these little bundles that myosin can bind, you know, and of course we've got things like, you know, rho. We've got things like rock, you know, bind into myosin and activating it and stuff like that. So, you know, this is just a little smattering. You know, anilin's probably here and other things are certainly here. You know, down here we have all of our talons and all of that stuff, you know, connecting all of this. So now we're at this like structure, and we kind of learned a little bit about cell migration at that point too, right? Cool. So then we're at that cellular structure level. And so it's like, all right, well, let's just kind of magnify that a little bit. And so now we're, gone from this single protein in the membrane to a large protein structure to an even larger protein structure. So we go to a dividing cell now. So we're looking at an entire cell. And we're looking at a structure very similar to a stress fiber, but now a stress fiber is kind of, it's just one of many. But a contractile ring that pinches a cell in two, like this is an enormous structure that's changing the entire, universe of the cell. It's splitting the entire cell into two. It's kind of a tall order that this thing is doing. And people used to think that these contractile rings were kind of like this, were one big unit, but really what they're made out of is all we're finding is all of these smaller, actually, little contractile structures like this. So rather than being these big, single, long filaments, they're all these little contractile areas. Okay. And the paper that we were focused on was looking at these proteins that are kind of like way, up at the membrane all around it. But, of course, the fave, everyone's fave protein, which is obvianilin, boom, was binding here. It's actually looks a lot like a myosin to me, but it's okay. They all are kind of myosins. And then, we'll have our myosin here. Oops. We'll have our myosins coming out here in this color. Boom, 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 little motors. And then we're focused on row, which I always draw green, and this gap protein. Gap shuts off row, so gap is red. Or, orange. That works too. Okay. And anilin helps recruit that, and this helps, reduce and inhibit. This complex helps inhibit contractility. Oh. Okay. So, boom. These are our first little 3 stories right here. Isn't that nice? We've got a protein in the membrane, piezo. And then we've got stress fiber, large structure. And then we got the governor of cytokinesis. That's right. It is me. I'm trying to do an Arnold Schwarzenegger impression. Yeah, I'm the governor of cytokinesis, yeah. I'm here to split your cell in two. You do not think I could do it? Governor. 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 Yeah, I'm the governor of cytokinesis. And I've come to hasta la vista, your obsession site. Cytokinesis. You have very kind nieces. Okay. That was that's really fun to me. I love this. The governor of cytokinesis. Which is, of course, P190. P190. And anilin. Anilin is chilling up on your cell cylinders, making them feel things that ain't used to feeling. I may or may not have written a rap about anilin. If you're lucky, I might do it for you. Okay. So those are our first 3 stories. Fun, 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 fun. And then our next story is kind of just an extension from that, which is, I think I might be able to just, oops. I'll just draw a new one. So, then we go into, actually don't. I remember what the next one is, but I don't know if I actually remember all of the papers, the order of them, which is okay. So we looked at the stem cell paper next, and the stem cell paper was a new paper to me, and I was really glad that I found that paper because it just kind of shows how important the this whole mechanics thing is. I didn't know it was so important for stem cells like that. I mean, I knew it was important for cell differentiation, but I didn't know we would, like, knew the mechanism of it. It's really cool, right? And so what they found, if you remember, was, we got our cell like this, and we got our nucleus in blue. If you never see anything blue, it's a good guess. You can guess it's probably a nucleus. Scientists just kind of assume people to know a lot of stuff. Whoa, they both blued up. You know what? I don't hate it, but I feel like this does have to be a different color. But I didn't hate it. Okay, cool. All right, and then our DNA, let's make it a lighter blue. Because, okay. Oops. Lighter. Nope, somehow we're stuck in this land of it not wanting to take it. Take it. I said take it. Okay. Great. So when the cell is under tension, its DNA is more curled up like this. It's more embryonic in nature. And the only way I can actually remember the way this works is because I remember this analogy. So under high force, stress, I'll do the arrows. I usually do force in red. Force seems like red to me. I don't know why. Why is it? Sometimes I don't understand. Illustrator. So under high force, and this is a stem cell. See, it's like only wants to take this is actually skin. Under high force, what's happening is we have actin accumulation, all out here. Again, we're not taking the color. Take the color. Thank you. Okay, so we have very little G-actin in the nucleus. And G-actin binds to these areas and kind of opens them up. There's other stuff going on too, right? So, but all the actin comes out of the nucleus because it's got to help kind of like build a robust support system out here with myosin and all these other proteins. So that's under high force, and then we could draw the low force version of it, just so that we can see that as well, what that would look like. We just get rid of all these, not all of them, but most of them. Goodbye, I hate you, I don't love you, or your families. You're all stinky and not worth existing. Okay. And then all that G-actin is now instead in here. Where it can help regulate and like silence big swaths of these DNA. And then maybe up here it's like yay, like that. So now all this can be expressed, and now this is like a differentiating cell. So let's maybe say it's expressing a bunch of keratin to make keratin filaments. Ha, I tricked you. Yeah, look at all this. I knew it wasn't going to be switched to green, and I switched it. Boom, so now we have a bunch of keratin being expressed because it's coming from woo 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 there. Cool. This is low force. Low force. Low force. Equals keratin. Oh, did you see that cheating? Keratin production. I'm feeling, I could type this out, but I feel like I'm committed at this point. Oh yeah. Okay, great. That's gonna feel good to be done with that. It's not real pleasant to write that way. Gonna be honest. Okay. But I wanted to be thorough with my labeling, so it makes sense. Cool. So, dang, that's 4 papers. What was our next paper? Paper number 5. I don't remember where we went from there. Let's take a peek. Nope. Let's see. Papers. Was it this one? No, that's the first one. Okay. That was the next one. Oaks. Oh yeah, it was Plotnikov, I believe. Yeah. Yeah. Yeah, this paper is cool. Right. It was on the tugginess of the. Yeah, I like this one. This one was a new one too. So this one we doubled back basically to this little area, to these stress fibers, but instead of just looking, at one single one and concerning ourselves with just that, we're kind of looking at all of them in a cell. And, how their dynamics are super, super important in like cell migration. I had no idea about this. It's really interesting, so they all have these like tons of these different stress fibers and focal adhesions. Let's draw one in each cell, but like, let's look at that one, that cell there, and then we'll take another one here. Oops. Copy. Yes, thank you. And do one more. Copy. Okay, so now we got these 3 cells. Let's move these down a little bit, actually, right here. Let's move this all over here. Let's do a little organization to make our lives better in the future. Okay, so there's our 4 little papers that we did, and then we'll do this one. And let's rotate these like this. Yeah, cool. Oops. Actually, let's do it like this. And then let's reflect that. Cool. Now the words are backwards. We don't need the words. It's fine. Goodbye, words. Goodbye, words. Goodbye, words. Goodbye, words. Okay, so we got these cells like this. And these cells want to crawl. But, we don't want them all to crawl different, or we don't want them all to crawl the same distances, because we want them to crawl different distances, because we want different cells to get to different places, just like humans. Everyone's got places they need to go and be. So do cells. Some cells want to go farther than other cells because we need those cells farther. Some cells need to go closer. So how do cells do that? Well, they touch their environment and they pull on it. Depending on how much they pull on it, like this cell can pull pretty good, it'll go a certain amount of distance. Let's say the cell it'll go about this far. And then it reaches some point where the amount that this can pull right here is equal to this amount. So we have, let's say, let's do our force. We have an equal amount of pull here. Woo! Or as I like to call it, yoink. And it's equal to the amount of yoink from the environment. So this is environment. Yoink. Put a V on there. Cool. So they're equal to each other now. You know, here, let's say that the yoink was, you know, this, like it was over there. Not the same. But over here, the environment only yoinks back this much, just a little bit. So it wants to go till it's equal, basically. That's what they found. They characterize all the different styles of pull and tuggy here to show this. Now this one, let's just start cutting away at some of this. Let's reduce this one. Reduce the tugging, right? Get rid of some of these, some of these. Alright, so that's the can tug less. You guys see where this is going. We're geniuses in this class. I know we are. Right? So this one's going to tug, can only tug a little bit. Can't tug very much. How's that look? Okay, that is, well, it's thicker. I think it actually, I don't want it to be thicker. Let's just make that one real small. So that's, you know, it's about the same size. So let's say it actually just doesn't move. It's happy where it is. You know, this is maybe it started further back. So it's about the same. So it doesn't move. All right, now this one, let's, you guessed it. Boom, let's increase that. Boom. Let's double up on some perps. Oh, we doubling up. Schnikes. Rice to nugs. Schnookums. Okay. Wheeze it. So now this, obviously, we know what's up. This is a party. This one, oh my, this is a party. A party! Can't compete. This can't compete. So, obviously, this one's going to go for it out here where these finally will equal each other. So, it's not a stick, but you get it. This one's going to stay put, and then this one's going to go there. Right? So it's a way for cells to kind of like navigate through stiffness. Stiffness. Make this easier on me. Stiffness navigates cells. Pretty cool. I'm into it. This one is I feel like this one, Piezo. We need some words about it. Piezo. I guess I just like mechanical channel. You know, I feel like that describes it. Or what if we write milk carton? I think that makes sense. We don't get what that means. Milk carton. Maybe we could draw a little milk carton, you know, for the kids. Dang, this looks like ominous. This is my cow. Okay. Okay, so that's the first 4 papers, or 5. So the next paper was an extension of this one, kind of, really. So what they did is they just took cells. The researchers took cells. They just plated them on different stiffnesses, right? So we got our 3 cells. And then looked at them over time. And these cells on soft. Matrices just turned into neurons, basically. You know, they looked at branching and other cell shape stuff to look at these. So this is on soft. It's a neuron. Oops. Actually, that worked. And this is these are stem cells. Okay. So then on an intermediate stiffness, cells turned into muscle cells. They turned into these like little, like spindly looking kind of cells like this, which are like a muscle cell. Muscle cell. Fun. Cool. I like the way that looks. It looks cool. Okay. And that's on medium stiffness. And then we have stiff, which is bone cells kind of look like this. They kind of look nuts. They kind of look crazy. They kind of look like this. They're pretty big. They can be real crawly, and they can have these long spindles like this. Okay. And this is on stiff. So this, these are matrix, yeah, matrix type. So this is like pretty nuts that like just the matrix alone can, you know, control what kind of cells we get. That's not what people thought. So this is like confusing to a lot of people, I'm sure. You know, this is like what? This blew some people's minds. For sure. Okay. Boom. Here we are. Let's keep cruising. This is fun. So I'm trying to grab this black background, but I have to get it right there. Why won't you let me grab you? Don't understand. new sublayer. And do this. Shit. Nope. Don't want it pink. Oh my god, it'll kill me. Cool. Great. Lock. Back on this. Great. Look at this. Cool. So, let me draw these actually. Cool. So, the next paper, that was 6, 7, was junctions, alpha-catenin. So yeah, so now we're exiting from just thinking about cells. So here we're thinking about a protein, then a protein complex, and then here we're thinking about, you know, a larger complex. This is actually, you know, not to scale in any way. This is actually smaller than this. And then we're thinking about how it affects the whole cell and its fate and its destiny and its ability to know where it's going and its fate and its destiny again for different types of cells. And so then we start looking at its connection to other cells to show that that's a mechanical process as well. So I kind of started drawing these up here, so I'll snag one of these. Right here, this will be our little secret. Don't tell anyone I drew that earlier. Shh, it's a secret. Okay, so now we get into, actually want to go back here, lock that, unlock all that, yep, pull all this down. So this is like where it gets fun, because we get into now we're talking about the structures that hold cells together, which is a fun thing to be talking about, not only because I have a PhD in it, but because it is, without it, we wouldn't exist. You know, this is what separates a single-celled thing from a two-celled thing. It's the ability for them to like hold on to each other. Okay, I've lost a lot of the parts here, but that's okay. So, you know, and cells just basically, whoops, just disappeared it. That was funny. I'm a magician. Oh, I'm on the wrong layer. That's why. So we have, you know, these different junctions, and they have different different properties, and actually, let me just, I went through this whole thing of stealing this. Let me just start over. It'll make it easier than actually trying to recoup that one. Okay. So we're in epithelial cells, and epithelial cells, let this be purple. That's too purple. I want it a little lighter. Sorry, I'm very picky. Okay, that'll work. So we've got our epithelial cells like this. Oop. Sometimes you gotta. That's this epithelium is not very consistently shaped, but it's all good. You know, sometimes they're a little goofy shaped like this. Okay, I like the way it's coming out now. Alright, and then we've got our junctions. We've got a couple junctions up here in this apical junction complex. We've got our tight junction. And then we've got our adherence junction. And these are up here, but they're also, you know, these cells are kind of far apart, but they also kind of decorate down here too. They're not as big, but these areas like where they're real close, maybe they could touch, like there. You know, I didn't draw these to scale or anything, but this one we can definitely show it much better. So like that. Maybe another one there. Okay, so those are adherence junctions. And then inside of all this, we've got a bunch of actin. just all over in these cells. Let's actually use these two as our little model. I like these two. They'll be our friends. You know, so all around the membrane, there's just cortical actin. We've talked about this before, and a lot of the papers have shown this. They're just all around the entire membrane. There's a little network of actin and other proteins as well. There's spectrin and things. And up here, there's just a frick ton. Like around the junction, there's these huge bundles of actin. And the organization here is really interesting in how they're different between the tight junction actin here, maybe. And the adherens junction actin here. And what their population sizes are. Like I kind of drew this one a little bit bigger than this one. And that's how it is in Xenopus. But it's usually the other way in a lot of systems. And flies, for example, they don't even have tight junctions. They have something different down here that does the same job of being this barrier, but it does it down here. It's called the septate junction. And so then at these little areas where we have these middle junctions, like this one paper talked about, there's usually a little bit more actin there. That's supposed to be actin, so make it more red. You know, they come out like that. There's some little nesting, basically, of actin around that. And that's its little actin egg, or junction egg. But it's nothing like up here. Up here there's just, you know, these, in comparison, are humongoid. Cool. So that's just the basic setup of actin, and that's all important for helping pull and mechanically integrate all these structures and forces and yanking and yoinking that happens between these cells. Additional junctions that we didn't really get to talk about are gap junctions. So gap junctions actually form pores between the cells. So there's something like this. Yeah, this is actually pretty good. They like kind of form these pores like this. And I think they actually bring the membranes together, but I'm not gonna go through all the trouble of doing that. So we'll just say it does something like this. And these are really important for that programming and stuff like that. They're really important for sending signals and patterning between cells because they form these gaps between the cells that allow them to communicate cytoplasmically. So, for example, if we have some of these bad mamma jammas, some piezos down here, for example, that start opening up. This was not in the paper, but I'm adding it. And these start adding in calciums. Whoa. Those could go from that cell into that cell. It's cool. And so you can get patterning of pulsatile contraction and things like this. Okay. So those are gap junctions. Then there's also these things called desmosomes. We'll do desmosomes over here. Desmosomes are really cool. We'll just draw them real big. They are real thick. Boom. Make this one even bigger. Okay. And these are kind of all over. They're down here. They go up here. And these are connected through keratin filaments. And they look really beautiful. And what these junctions do is they really mechanically integrate the whole cells in all of the tissues. You can see how they do that. It's basically like forming this like spider web. I thought that was a junction up there. And you can see how having all the cells have these. Let's say there's not going to be one up there, but you can see how that it just forms this meshwork of cytoskeleton elements to distribute all the load and all the forces around. But up here we have these specialized little centers that are these mechanical signaling centers that integrate the apical surface of all of the cells, because all of these connect really with actin all around, just like a contractile ring is made up of all of these little contractile units like this. If you look at the cell from the top down, it's the same way. So if we draw this cell, and I talked a little bit about this. Why? Okay, so there, just do that. Okay, so if we draw these cells like top down, I talked a little bit about this in lecture. They form these contractile units of actin, and people have. This is actually what people first saw, but they didn't really notice it and pay attention to it until later. They actually form these little contractile units like this, just like in cell division. If you think about this, this is exactly like a dividing cell, just cut sideways. We're just looking at it in a different way. So, this, it'd be like if we were looking down, we'd see this actin like this. If that makes sense. It's like flipped. Rotated. Okay, so what they found in this is they were looking at these proteins that were attached to the cadherins. And they showed that this protein, alpha-ketene, is kind of folded up like that. And that's how it attaches to actin. So when it's just chilling or whatever like that. And then they showed that under higher tension, what happens is alpha-ketene, just straightens all out. This allows this protein, vinculin, to bind. And so now both alpha-ketene and vinculin can bind to actin. And vinculin also is important for contributing to bind to other things that help polymerize actin to make even more actin too. So not only do you get, just look at this. You get this little kind of bendy straw that's chilling to a bendy straw with now an anchor and all this other stuff. It's just a way to reinforce the cell and distribute that. So if we get a bunch of forces, we get more reinforcement between the cells to connect things. So this connection between the cells is a sensory organ. And so now the next paper that we looked at was taking this idea of cell-cell junctions. Sorry, I gotta get a drink of water. I was collecting my thoughts. Okay. So the next paper is looking at these endothelial cells, which line our blood vessels. And I gave you that little paper about, and that whole sort of explanation of those. So we don't need to go super in depth on those. But they found that when we have, low flow versus high flow, there's these two different states for this notch signaling thing. Mechanism, this whole butterfly new mechanism versus high flow. And I'm going to make this green. And I'll tell you what, I'll tell you something. You better listen. Make it all a little smaller. Because then everything makes sense. Okay. Through the notch signaling, the transmembrane domain of notch, the cells sense this flow and they kind of, seal it up. So under low flow, cells are in the endothelium they're kind of like this. There's some ability for these cells to have some leaks. Sometimes they might be sealed up pretty good. Oh, it's kind of like a little bean. And so we can have that leakage that happens. Oops. Of blood and cells. Ah, beautiful. And the other one, look at it. Do I still have it drawn up here? I think I do. Haha, yes. Look at this. I'll just grab this whole thing. Grab this whole thing. Silly me, I already did this. Boom. Whoops. Stop it. Quit being... Just quit being mean. I'll listen to you eventually. Okay, let's just get rid of this because I already drew this out real nice. So let's just look at this real quick together right now, and then we'll move on to the next little part. Okay. So this here got the high flow and the slow flow. So under high, and we'll flop these because I think that makes more sense. So under high flow or slow flow, we got the ability for this stuff all to leak out. Because the cells are not as sealed up. They don't crawl together as much. So we got these openings so things can leak. Under high flow, it's all tight. You know, this is changing all of these dynamics in our vascular. Here we have high flow, so it'd be sealed up. Here we have low flow, so that it can be more open. And this works not in the canonical notch signaling way, where we have this transcription factor, but by having the transmembrane domain break away, bind to E-cadherin, scaffold all this branching of actin to crawl all these endothelial cells together. Really cool stuff. Really in-depth SWIT pops. Really cool stuff. So this is this one over here. Let's label it. This was alpha-catenin. Alpha-cat is mechanically sensitive. Kind of like that. Sensitive. Sweet. This is vein cells. I like that. Vein cells. Feel flow. And seal. They really love seal. Kiss from a rose on a grave. Do da dee do da be da dee da dee da do. I don't know the words to that song, but that song was the shizzle when I was in middle school. Let me tell you. Okay. So that was the last paper. Then we talked about my paper just a tiny little smidgen. So indulge me while I talk about that again a little bit. And what I've basically looked at in those cells, I'll go through this real quick, is in control cells, anilin knockdown cells, and anilin overexpression cells. What anilin does is it doesn't affect just this actin here. Control cells, there's good amounts of actin. There's also a decent amount of actin all up in here. And when you knock down anilin, it's like, oh, it messes all this up. But really, it messes kind of all this up too a lot. And then this, when you overexpress anilin, oh dang, this stuff goes nuts. The junctional actin goes nuts. Sure, yeah, it sure does. But this stuff is really freaking nuts. This stuff forms like huge bundles like this that span the membrane and deform it and are pulling on it. These basically huge apical stress fibers up at the top of the cells. Because remember, we're looking just up here at the tops of these cells is what we were looking at. Okay? So this is, sorry, anilin knockdown. I'm sorry. This is wild type. And this is knockdown. So these first cells here are just control. Just to be clear. No modifications. And then this is overexpress anilin. And that causes all those changes in force. And the way that I looked at that force and all that stuff was by doing things like making like laser cuts. Like, oops. Making laser cuts like that and measuring things like recoil. And the next paper uses that technique quite a bit. Paper that you're going to read is from Adam Martin's lab. And Adam Martin is one of the first people who really showed the importance of this medial apical population of actin here, like that. The stuff in the middle. He showed that's really important in Drosophila for ventral furrowing, and I've shown some videos of this. But this is the paper, this 2009 paper, and it really influenced my work. His work did, for sure. You know, he's doing apical cuts here and showing it's under recoil. You know, looking at myosin, showing that it's accumulating in the center of these cells, and this is what's allowing these cells to contract. Yeah, medial, junctional. I wonder if there's a cool video of his that we could watch. Ventral furrow. Yeah, let's watch this. Boom. Okay, so this is that. Yeah, this is the video that I want to see. So you can see all these in green myosin pulses accumulating on the medial apical surface, and it's those pulses that accumulate. It's those pulses that contract the apical surface of the cells. So as this apical surface contracts, it's pinching the top of that cell in. And as all of the cells pinch the top in, it basically causes the tissue to buckle right there and fold in on itself. So I can draw that out a little bit. I'm losing my mouse cursor. There it is. Oh, okay. So if we yoink these two cells, actually no, I'll just draw them out. Yeah, let's just do it down here. So that's not the color I want. So if we have cells like this, and they all contract their apical surface inward, what that's going to result in is an apical surface something like that, an apical surface something like that, and an apical surface something like that, and then like these kind of like big, like bulging cells, right? Because they've lost a lot of that apical surface area, has to go elsewhere. And that causes the tissue to bend like that. So you can see how that'll bend the tissue if you have a lot of cells do that. But it's interesting if we would just watch this if we watch this a couple more times. Can I speed this up actually? I like it to be a little faster. you can see that it accumulates the myosin, just right in this band right here. It's not really accumulating any out here or out here. And so how does it, it does a little bit, but how does it know? And how does it coordinate? How do these cells coordinate that? These are cells. How do they talk to each other and communicate to each other through forces? Well, we kind of know. And that's what this paper is about. It looks at the catch bond aspect of myosin 2. And it shows that, you have this like, let's make a little cellular patterning. Oops. Can't zoom in anymore for some reason down there. I think it's because I'm at the edge. Fine, whatever, fine. I'll take my ball and go play elsewhere. Great. Don't need you. I don't need to be down there. I want this stroke to be. I just want to fill it. Let me fill it. Yeah, cool. Okay, so let's make a little epithelium here. It's fun. Look at that. Doesn't have to be perfect. You know why? Because biology isn't perfect. Okay, and now if I do this, I will be in business. Sometimes I'm smart, but sometimes I do miss the mark. I could have done this much smarter. But I'm having fun. It really doesn't matter because I can just go like that now that I think about it. Okay. Okay, well I'm just making a solid color here, so this makes no sense really. But I could do this. Yeah, so let me do this actually. So somehow in here, we're accumulating, and only in here, in these cells, the myosin. And the way that they do that is myosin likes stiff things, just like a finger trap. If we have our little fingers, and we have our finger trap, this thing loves to get pulled on, right? It pulls on tighter the harder you pull. It pushes down to hold on tighter. And myosin is the exact same way. So myosin starts accumulating in these little cells. It starts accumulating here and here and here and here and here and here. Due to signaling in those cells. Upstream signaling from, I'm not sure exactly, it's from the last paper, I don't fully remember the whole mechanism of it off the top of my head, but our very last paper from Eric Weechhouse shows you how these junctions are stabilized during this process, and there's twist and snail are being expressed here, so there's some developmental cues, and those developmental cues kick off these mechanical cues of accumulating myosin, and then there's this positive feedback now, because when you have myosin, what do you have? You have, of course, actin structures all forming along, all in here, connecting all of these together. And what happens when you have a lot of high actin levels? When you have high actin, you have increased stiffness, right? Boom. And so we have increased stiffness, and myosin is a finger trap, right? And so it likes stiffness. So then, what do we get? We get more myosin accumulating here. And more and more and more and more and more and more stiffness and more actin and more stiffness and more actin. And you can see how we get now this just positive feedback loop that then builds all that stuff. So they don't really talk about the myosin power stroke. I don't really think much, or I'm sorry, the myosin's catch bond. This activity, like this is like a catch bond activity. Catch bond means it becomes stronger gripping, or its grip holds tighter when you pull on it or when it's pulled on. Myosin catch bond. Coordinates. Coordinates. Does it coordinate? It's like it communicates the signal of myosin self-accumulation. It's coordinating. I think what we'll say is stiffness. Stiffness. So just like how here, stiffness, environmental stiffness. Where is that? This one. Dictated how far these cells can go. Here, environmental stiffness, basically, the stiffness of that the junctions are feeling against, so the stiffness that this junction feels on this junction compared to this junction on this junction is different, right? And it likes to pull more here, so it accumulates more myosin, and by accumulating more myosin here, along these paths, that allows the ventral furrow to form. Does that make sense? Yes. So, the junctional stiffness, the environment that this junction feels here compared to this one, is higher. And that helps dictate and control the myosin localization. So it's almost, but it's almost the exact same thing as the focal adhesion one. But it's, instead of regulating migration, it's regulating the orientation of accumulation so that we get folding of a tissue. So it's this balance of force. And when we have high stiffness, we have high myosin accumulation. So let me just write that. High stiffness equals high myosin. Okay? Which regulates, and high myosin means furrow formation. We'll just leave it at that. I had to do a lot of talking it out to get to that little set, but that makes a lot of sense. It's pretty cool. In the discussion, I'll have a shirt, and you can kind of see this process live with just a piece of fabric. If you pull it, you can see how when it gets stiffer, it just kind of wants to furrow right there. It's cool. So then, from this paper, what we then are going to look at is we're going to look at this is an Adam Martin paper, and Lance Davidson is on this. And Lance Davidson studies Xenopus embryos like I do. And he was really influential in me thinking about them as little mechanical things. He always talks about, so this is a Xenopus embryo. And this guy, you think I'm a nerd. This guy is a Xenopus laevis mechanics nerd. So Xenopus embryos kind of look like this. They got that big yolk at the bottom, and then at the top they have a lot of pigment usually, if they're pigmented wild type frogs. And so this is a single cell at this point, and this divides a bunch of times. I've shown you some videos of dividing frogs. And he always talks about how if this was a beach ball, it would be so fun to study because it'd be so big you could easily pull on it and push on it. So like one of the things that he does to study it in my paper is you just like cut off a little part of this and you just push on it with a device. You can measure it. Or we'll take another device that looks something like this. Let me actually like zoom way in here. Let me change the color too. It's just got like a little tube like this. Real small. Like, a little device. And then this whole thing like hooks up to like this big machine or whatever. And that just sucks on this, aspirates, sucks on this, and you can measure how much the force is and all that stuff. So it's a push and pull on embryos, basically. And so what they found is that as this develops over time, so instead of it looking like that, it would look something like, I don't want to draw all that out, so I'll just do some little detail. We got a little tadpole like this. They have these little curly intestines that are really beautiful, and their heart is really beautiful too. Okay, so to go from this to this, basically, what they found is that the whole embryo gets stiffer. So that's the next paper. And we also have another paper that we're going to do alongside that, which kind of goes back to this ventral furrowing stuff. And it shows that the stiffness is really important for regulating what's going on here in a different way, showing that it's really important for maintaining cell-cell junctions. And the reason why that's important is, I think, is because of cancer and all this other stuff. And we'll go into all of that maybe in the lecture video, just so that you guys have that as well. So I don't know if you can see this, but this is a lot of different stuff. But it's kind of nice because it's a lot of different stuff, but it all is connected. And I didn't really do any of this on purpose. So other than, at the very beginning I said I just wanted to start at the membrane and build up from that. So we did that. We started at the membrane, and then we're into whole embryos by the end of this thing. Whole organisms, right? And from this, some interesting things emerged. We've been talking a lot about emergent behavior with the notch signaling, for example. And this is an emergent behavior, right? It's from a few simple rules we get this complexity. We can get bird flocks and schools of fish, herds of sheep, patterning of hair cells through notch signaling, right? In Minecraft, we could do redstone and get simple rules can create these patterns, right? And that's basically emergent behaviors. Like, what is the behavior that emerges from these simple rules? And we've done that several times in this class. One of the examples of that I didn't really get to talk about much, but we did it, and we built towards it, is that improv where we were listing off counting off our numbers. What really are the rules of that? It's a few simple rules, and then we have this emergent complex behavior. At this point, all I have to do is say, ask a question, and then you raise your hands. All I have to do is say, count them off. And then we count them off, and we know what that means. We know that means that many people had that question, or had that same question, or thought that was interesting, or whatever it means at that time. It's really interesting. And another thing that happened is all of these emergent connections that happen just because we're staying in that same world. So all I did was pick this as my starting point, and I knew my end point I wanted to be somewhere around here. But because of this, look at all these different stories that we have. We have a single cell piezo can open up, and that could affect the mechanics of these stress fibers. And these stress fibers can impact the forces on the nucleus. They can affect the forces of the cell's ability to navigate, right? And this, the forces of these cells on the differentiation, they affect division as well. So we have a cell dividing, and then we could have a cell, sensing some signal. It wants to divide because its forces outside of it are changing. And then it wants to then start migrating away, where it's going to become a different cell type. It's going to merge, right? Maybe join up with some other cells in a tissue and form cell-cell junctions, and sense the mechanics of that cell-cell junction, and properly signal and build them in that way, and connect those forces through its other neighbors, and they have to be balanced. We have too much or too little, it affects the force balance. And that force balance is not only important across cells, but it's across tissues. And the whole embryo is stiffening over time, and that helps cells guide themselves and become different cells and differentiate. And that's really important for maintaining homeostasis in cancer and things, potentially, which is what we'll see and talk about in the very last paper, set of paper. It's not directly related to cancer, but it's about twist and junction formation here. So you can see we kind of made this whole little story about stem cells. So if we go back to these little drawings here, if we go back here, and I want a little space, let's see, let's do it over here, unlock this. Okay. Lock her up. Boom. Let's say we're in an intestine. So this is, let's say our intestine. Actually, you know what? I actually have, let me go. I have a nice epithelium that I made. Let me see if I can find it. Old labs, Miller lab, figures. Boot doot dit 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 boot. I don't think I'll be able to find that. No, that was a couple years ago I made that. Okay. Where'd that go? Okay, let's grab a new one. Okay, so in your intestine, so for example, this whole story of all of this data can be happening in a single cell throughout its life. So for example, in your intestine, if you're not familiar with the cellular architecture of your intestinal epithelium, very simply, this is not the best way to represent this, very simply, it's a series of peaks and valleys. I think I'm at the edge there. Scoot this over. Let me make these a little steeper. And the reason why your intestinal cells do this is because it increases the surface area of your intestine to absorb nutrients, right? Hills take you a longer time to walk over because you're walking over more land. You got to walk up and then down when you could just walk straight. So they're like this. And this is, they're very big and large and beautiful, but this increases the surface area. And down here in these things called the crypts, so this is your crypt. Crypt. I don't know what these are called. Oh, these are your villi. Villi. Okay. Down here, these are stem cells. And they're like a little conveyor belt. So these cells divide, so they form these actin structures. And they cut themselves in two. They're going to be under different forces and things like that too normally, right? So they're going to cut themselves in two, and eventually they start migrating along this chain because the cell's going to divide and it's being pulled on and tugged on by its neighbors here. So it's going to differentiate and become a different type of cell, you know. So we have cell division here. We've got migration and stuff. It's not really this type of migration, I don't think, so much, but you get the idea. And then up here what happens is these cells actually later they start to go like this and they break away their junctions and they fly away and get sloughed off and go on a ride out in the intestine and get digested and go away. We just chew them back up and eat ourselves. So they get extruded. That's what that's called. Extrusion. Boom. So you can see a lot of these. Here we have junction disassembly. We have coordination across tissues. We have stem cell differentiation. We have cytokinesis. We have signaling between cells. We could have changes in the mechanical environment as this goes. You know, right here we could have down here at these troughs. Let me draw them over here. Let me draw it over here. Yeah, this will be good. Down here we could have very soft matrix. And then up here, you know, maybe it gets a little more rigid. This could help differentiate the cells. And then up here, we have very rigid matrix. So as the cells progress up here, they're going to differentiate more. And it's going to change the internal signaling inside of these to maybe eventually be like this and get sloughed off. So you can see how this whole little mechanical world that we thought about can be applied to a number. This is just one little example about how this knowledge can be applied to the cellular world, and how you could think about it, you know, in diseases. So like any one of these mechanical processes is potentially important, you know, in a process like this. And so you can really kind of search for, if you're interested in the intestine, like intestinal-based diseases, then you can start thinking about, okay, cell-cell junctions are going to be important here, and you know, differentiation and all these different things. So yeah, it's kind of cool how this all came together. I didn't plan that. I didn't plan for it to come together in this kind of like little story about like a stem cell and maybe it crawling around. You know, there's all, you know, stem cells are dividing and crawling and changing everywhere in our body, not just in our epithelia. So yeah, it's cool how, you know, from this simple rule of just I want to start at the membrane and go to embryos, out of this emerged this sort of whole little story of the cell in these little parts, and it's pretty cute, and I'm very glad that we all got to share it together. This is that little thing we did together. I don't need that now. So that's really it. I hope that was useful and you like that. I'm gonna organize this a little better and I'll post it for us. So that's really all I had. If you have any questions about this, let me know. I'm happy to answer any of them. Yeah, I think that pretty much covers it. So have a good evening, and I'll see y'all on Monday.